A very good afternoon to everyone. Let's begin with today's session. And as we say in the starting, that let's crack CSIR-UGC NET with Unacademy. So the topic of today is origin of cells, and this is the part 2 of the continued lecture which I took in the previous part. That was the part 1. This is the part 2, the lecture, that is origin of cells and unicellular organisms. So before beginning with the topic of today, let us have a small introduction about myself. I'm Farida Bandookwala and I'm pursuing my PhD from DAVV. I've done my BSc, MSc and MPhil in biotechnology, and I've qualified MP SLET examination with a teaching experience of more than 10 years. So what you will get with the Unacademy is that you will get daily life classes so that you won't feel like you're not connected with the curriculum. You'll very much feel motivated each and every day because you'll be getting all the upcoming exams. So there are daily life classes, and there are structured courses, and you can take these courses and you can start studying at the comfort level of your home. Then there are live tests and live quizzes, so you attend these live tests and quizzes and you make the most out of it. And I always say that it is very important for anyone who's preparing for a competitive examination to evaluate how much they have learned so far. So that is possible with the help of tests and quizzes. Then unlimited access is another added benefit of Unacademy. Then you can learn anywhere, anytime with the help of Unacademy. Learn anywhere, anytime. If you're not in the town or you're not particularly available at that particular time, you can just watch the pre-recorded videos. Like once it is live, it is always there on the platform. And there are live classes, and what is so good about live classes is that while the class is going on, you can ask your doubts and queries directly with the faculty. They'll be very much willing to solve your doubts and queries while the class is going on live. We take up the questions as soon as you post it. So it is one of the very great benefit of live classes. You can just go to Play Store and download the Unacademy app, and when you will have downloaded the app, you can make your account, you can log in, and you will have access towards the special classes. And these special classes are conducted on the platform of our app, that is Unacademy app. And the free classes are conducted on YouTube as well, which we are doing now. So all the classes which are on YouTube, you can anytime access them, and these special classes also access them without paying any amount. And this plus subscription is paid subscription, and on this plus subscription there will be short-term courses and crash courses for all the upcoming exams, and you need to pay a certain amount for getting this plus subscription. And what I would suggest is go for a 6 monthly or a yearly subscription, so that it can be very much beneficial for you, so that you can make the most out of the available content. And in this subscription charge, which is quite pocket friendly, you can still get a discount of 10% if you're using my referral code, that is FP01. So do not forget to use my referral code FP01 and avail 10%. So let us begin with the class of today, and the topic of the class is origin of cells. Origin of cells and unicellular organisms. This is the topic, and this is the part 2 of the series, and we have already seen the part 1. In that part, we have learned till the formation of proteinoids. Proteinoids formation done so far. What are proteinoids? They are branched chain of proteins. Branched chain of polypeptides. So proteinoids and proteins, if a question comes, what is a difference between protein and proteinoids, is that proteins, they are completely active, they show various functions. And what about proteinoids? Is that proteinoids, they are branched chain polypeptides and they are not completely active, though they show some kind of catalytic property also, and susceptibility to digestion also. These all properties are exhibited by proteinoids, but they are not complete proteins in nature. So this we have seen the last class, the formation of proteinoids, how it occurred, and we have seen the Allen and O'Brien experiment, we have seen Stanley and Miller experiment. These two experiments we have seen. Now from today we have to talk about coarser weights. Coarser weights. This is the topic, um, or the beginning topic of today's class, that what are the coarser weights. So the term coarser weight was actually first of all introduced by the scientist O'Brien. O'Brien gave this term, coarser weights. Now, what are coarser weights? They are actually aggregation of colloidal particles. Aggregation of colloidal particles. And if you are saying colloidal particles, so that means that they are in the liquid state. Colloidal particles in liquid phase. And this liquid phase, it persists for a long time as the suspended droplets exists in suspended form. Suspended droplet form for a long time. So O'Brien, he gave this term, and they are aggregation of colloidal particles. Then what else? Formation of coarser weights occurs when the dispersed colloidal particles separately and spontaneously they merge into a solution and droplet, and they form a special condition, in which temperature and acidity, that is like appropriate for the formation. So coarser weights, they are also exhibiting some kind of property. So formation of coarser weight, when does it occur? Formation of coarser weights. Formation of coarser weights, when does it occur? When dispersed colloidal particles, they separate spontaneously out of a solution into droplets because of the various conditions. So imagine that this is the solution. This is the solution, and out of this randomly some droplets, they separate out. Droplets separate out, and this separation occurs because of special condition of acidity and temperature. So separation of smaller particles from a large solution leads to formation of coarser weights. Now when we are talking about coarser weights, there must be some special property of coarser weights. That's why we are talking about it. So properties of coarser weights. What are the properties of coarser weights? They are able to exchange the substance with environment, ability to exchange substance with environment. So that is not a closed compartment. It has got ability of exchange. So ability to exchange the substance. Then it can increase in size, ability of expansion. Then it can selectively accumulate certain kind of compounds in them. So it has got the ability of concentrating certain compounds in certain compounds in them. So these are the properties of coarser weights, that it can exchange material with the environment, it has the ability of expansion, and it can concentrate certain compounds within them. So this was about coarser weights. After the formation of coarser weights, we should move towards microsphere. But you know what is an important thing associated with coarser weight that we should write over here is that the coarser weights, they are non-membrane bound molecules. The most important thing that you should remember for the exam point of view or for attempting the question, so they are non-membrane bound. They are non-membrane bound thing that is associated with coarser weights. Now talking about the next thing, that is microspheres. Microspheres. So what is the meaning of micro? Means small, and spheres means something rounded. So they are the molecular aggregation of proteinoids. So they are the aggregates of proteinoids. Aggregation of proteinoids is microsphere. Then what else about these microspheres? So like in coarser weights, we have just read that coarser weights, they are non-membrane bound. The thing that is associated with microspheres is it is membrane bound. Membrane bound. Most important thing. So the first membrane bound molecule to appear is microsphere. Membrane bound. Okay. Now let us move the story ahead. So there was a scientist, his name was Fox. So what did he actually told? That the microscopic membrane bound spheres are formed because of the aggregation of proteinoids. And when does this aggregation of proteinoids occur actually? So the aggregation, it occurs when the environment is quite in a high temperature. It is kind of a boiling soup, and when this soup actually cools down, there is formation of microspheres. So what does this Fox says? Formation of microspheres from proteinoids in boiling soup, and then cooling down. This was the uh, this was the idea proposed by Fox. So the microspheres, they are actually uniform in size, then they are stable and double membrane bound. So what are the properties? If talking about the properties of microspheres, properties of microspheres. So they are uniform in size, uniform in size, then they are stable, then they are double membrane bound, uniform in size, stable, double membrane bound, and appear somewhat like cell, will appear somewhat like cell, appear like cell, and it will also show certain qualities like cell. So exhibit properties like cell. So what properties like cell it is going to exhibit? That is, it is going to show certain fission and budding property. Fission and budding property. These two properties it is going to show. And some of the properties like original cell also it is going to show. So properties like cell. Properties like cell in microspheres. So what are the properties like cell it contains is osmosis. Then it is going to grow in size. And selective absorption. Selectivity in absorption of chemicals whichever is around them. So properties of microspheres, it is going to show properties of osmosis, growth in size, and it is going to show the property of selectivity in absorption. So all these things it is going to exhibit its microsphere. Now another thing that is quite similar or to original cell that we need to see is the spontaneous self-assembly of macromolecules into coarser weights and then into microspheres that indicate that occurrence of similar structure under the primitive condition would probably give rise to more organized structures such as protocells. So the next thing that come into being is protocells. So we have seen coarser weights, then we have seen microspheres, and now we are going to see protocells. So protocells, what are they? They can reproduce. They can reproduce. Then it can be subjected to natural selection. Subjected to natural selection. And another thing is that it will cause reactivity and formation of structure. So which would act as creative force, successfully enhancing the probability of advantageous molecules, reactions and structure. So they can reproduce, they can be subjected to natural selection and various chemical reaction. Various chemical reactions. So this we have talked about microspheres. Microspheres, they are membrane bound. Coarser weights, they were non-membrane bound. And lastly, protocells. So the word itself is saying that it was a prototype of cell. It is protocells. Now let us talk about that appearance of first membrane bound cells. Cells with membrane. So this was the prerequisite for the development of life. We want our life to be successful, it should have cell along with membrane. So this was the most crucial event, formation of the first cell with a membrane bound structure. So what do we actually need for membrane to be formed? We need amphipathic molecules. For any kind of membrane, we need amphipathic molecules. Amphipathic molecules. And these amphipathic molecules, what do we mean by the word amphipathic? Means they have got hydrophobic part and they have got hydrophilic part. These two parts contain hydrophobic as well as hydrophilic. So when these type of molecules, they are left in a solution, what do? The hydrophobic part, it goes inside, and hydrophilic part, it occupies the outer space. So this is how amphipathic molecules are very important in development of cell. So amphipathic molecules of appropriate size and shape, they would spontaneously aggregate. Like previously, they would aggregate like this in the water. So this is your meso. And when 2, 3 different aggregates of the amphipathic molecules, they are going to come in contact, they are going to form bilayer. This is how the amphipathic molecules are going to form the bilayer. And in this bilayer, how it will be? It will be continuation of a certain molecule, and it will form a closed vesicle. Closed vesicle. And these closed vesicles are going to contain certain aqueous solution inside them or certain other chemical compound in them. So creating a small closed vesicle whose aqueous content are isolated from the external medium. All the present day cells are surrounded by plasma membrane consisting of the amphipathic molecule, mainly phospholipids. So example of the highly used amphipathic molecule in nature is phospholipid. Phospholipid. So as soon as in nature there was a long chain of carbon, and from that long chain of carbon, whenever lipid arise, phospholipids came into being, and then the outer membrane of vesicles they have formed. Now the first membrane bound cells whichever was formed, and this process was spontaneous because of the phospholipid molecule from the prebiotic soup. So enclosing the self-replicating mixture of RNAs, we told that it will contain certain kind of aqueous solution in them. So it is going to contain what else in them? So like this kind of membrane bound structure would be there, and it would contain RNA inside it. It is going to contain RNA inside it. It is not clear that at what point in the evolution of biological catalyst and protein synthesis first occur, but in this case, whenever RNA was enclosed in a membrane, it would begin to evolve. Whenever this kind of structure was formed, then from that part or that point, it started to evolve. This was the point when evolution started. And then various other things were used, and from these the actual living cell was formed. Actual living cell came into being. So these all things, they come under the origin of cell and unicellular organisms. Now talking about the recent theories of the development of cells. So contemporary idea. Contemporary idea of evolution. Evolution of actually unicellular organism or unicellular cell, in context to that. Otherwise we know that how evolution has occurred. So this is in context to what? This is in context to our special thing, that is our cell. So what does this contemporary idea actually tells about the present time? So the contemporary idea, just a second, let me just, yeah, okay. So contemporary idea that there is no actual standard model for the origin of life. But the most accepted one in today's time is one or two theories, and these are actually based upon various number of discoveries. So now we are going to talk about those in a very rough manner. So what is it? What is it? The first contemporary idea, what does it say? That the prebiotic condition actually resulted in creation of certain basic smaller molecules, and those smaller molecules were like amino acids, and they were sugar molecules. So this was actually demonstrated by Miller and Urey in their experiment. So actually the prebiotic condition, or the time when actually life was not there on earth, that condition actually triggered the formation of life. So prebiotic conditions triggered prebiotic conditions triggered the formation of basic simple biomolecule. Then second idea is of the phospholipid, that it can form the bilayer. Phospholipid made the bilayer. Phospholipid formed bilayer. Phospholipid form the bilayer, that is the basic component of any kind of cell. Without a cell membrane, a cell cannot exist. Then another thing that came into being was polymerization of the self-replicating molecule. So self-replicating molecules, self-replicating molecules such as RNA polymerized. So it is believed that the initial period was actually of a period of RNA. Was a time when majorly the thing that existed in the environment for the continuation of life was RNA. So RNA. Then there was selection pressure for the development of the molecules with catalytic efficiencies. So it actually triggered formation of various proteins, and then they actually combined with RNA and formed the ribozymes. So need of catalytic enzymes gave rise to ribozymes. So there was a need of such kind of molecule. So ribozymes, they came into being. Then the most dominant biopolymer, the work was taken up by protein. Protein, the most dominant protein, the most dominant biopolymer. These all are some of the contemporary ideas of evolution, that how evolution must have occurred. Then let us move a little further in this topic, and let us talk about the actual evolution of cell in terms of the time and everything. So actual evolution of cell. Cell evolution. So the earliest cell, it dates back to the time 3.5 billion years. 3.5 billion years earliest cell. Earliest cell dates back to 3.5 billion years. So where does this cell actually arose? It arose in the primordial soup. Cell originated in primordial soup. So primordial soup, it actually originated, and that time was not actually photosynthetic. That time was actually chemoheterotrophic in nature. Chemoheterotrophic in nature. That particular time was chemoheterotrophic in nature. So primitively the organisms, they were heterotrophs because the environment was chemoheterotrophic. So primitively heterotrophs, they gradually acquired the um, capability to derive the energy from certain compounds of their environment, and to use their energy to synthesize more and more of their own precursor molecules, and thus by becoming less dependent on the other molecules for their food. So a significant evolutionary event was the development of photosynthetic ability to fix carbon dioxide into more complex compounds. So develop the quality of self-replication. Then the cyanobacteria, they are actually the descendants of these early photosynthetic oxygen producers. So chemoheterotropic type of nature was converted into oxygen containing atmosphere. Oxygen containing atmosphere. Then what other things which are related with this is the first eukaryotic cell appeared. First eukaryotic cell appeared 1.5 billion years ago. This is the breakthrough point. Then the first cell, it acquired more and more DNA and acquired the mechanism to fold these DNA. Second, there was intracellular and large gap in it. So we are going to see that in a second. Sorry. So yes. So finally, in this evolution, what are the three major events which occurred? So the first event was the cell should acquire more DNA. Cell should acquire more DNA, and then this DNA should be folded in a compact form. Then when DNA was in folded form, DNA should fold on proteins. DNA should fold on proteins. So this is the first event for the evolution of a first cell. The second event, is formation of membrane bound cells. Formation of membrane bound cells, as well as formation of large and intracellular membrane-bound organelles, membrane-bound organelles. So this is the second event. And then lastly, the third event should consist of formation of formation a prokaryotic of a photosynthetic cell, formation of a photosynthetic or autotropic cells, of autotropic cells, formation of photosynthesis, synthetic or autotropic cells. So, and in the third event, formation of eukaryotic cells, formation of primitive eukaryotic cells, which are capable of photosynthesis or which are capable of further leading the life in the normal manner. So this is how the events of the whole process of development of the unicellular organism it occurs. So let us have a very quick revision. So origin of cells in the unicellular organism. The first is in the last class we have seen the formation of proteinoids. And what are proteinoids? They are the branched chain of polypeptides. They are different than proteins because they are branched. They are called as proteinoids, and they have got certain functions of catalytic properties and all. Then the second thing that we should read is coarser weights. After proteinoids, coarser weights they came into being. They are non-membrane bound structures, and the word coarser weight was given by Oparin. And what are coarser weights? They are actually aggregation of colloidal particles in liquid phase, and they exist in the suspended droplet form for a very long time. So how have they emerged? Actually, there was a solution, and from this solution, when the droplets they separate out because of the acidity and temperature in the environment, they lead to formation of coarser weights. The properties of coarser weights is the ability to exchange the substance with environment, ability of expansion, and ability to concentrate certain compounds within them. Then next thing is microspheres. They are the membrane bound things. Then aggregation of proteinoids gave rise to microspheres, and the term microsphere was given by Fox, scientist Fox, and he told that formation of microsphere from proteinoids occurred in the boiling soup, and when the soup cooled down there was development of microsphere in the environment. Now, what is the property of microsphere? That is uniform in size, then it is stable, it is double membrane bound, and it will appear almost like a cell. This is the property of microsphere. Then it will exhibit properties like cell. That is the most important thing, that it will exhibit fishing and budding properties. It will exhibit the property of fishing and budding in them. So properties of microsphere are osmosis, then growth in size, then selectivity in absorption. It will selectively absorb certain things, and certain things it is not going to absorb. Then the formation of protocells from microspheres. They can actually reproduce. They are subjected to natural selection, and they are subjected to various chemical reactions. Then some cells with membrane form, and that is because of the help of various empipathic molecules which have got hydrophobic and hydrophilic part. So example is phospholipid, which is actually the present day membrane. So because of these empipathic molecules, closed vesicles they form, and inside the closed vesicle, first thing that actually was in the bounded form was RNA, and that is how the cell evolved, and RNA was selected, and there were many changes in RNA itself for the further development. Then let us talk about the contemporary idea of evolution. Says that the prebiotic conditions it triggered the formation of basic simple molecules. Formation of phospholipid bilayer must have occurred, and self-replicating molecules such as RNA must have come into being and polymerized. and the need of catalytic enzymes such as ribozymes is there, and protein is the most dominant biopolymer in this present time. This is the thing. Now talking about the cell evolution, it actually occurred 3.5 million years ago, and they originated in primordial soup. Then chemoheterotropic condition was there. It developed the quality of self-replicating molecule. Oxygen containing atmosphere was developing, and first eukaryotic cell appeared 1.5 billion years ago. And the first event for the development of the unicellular organism is cell should acquire more and more DNA, and this more and more DNA should be kept in a very systematic manner. The DNA should be kept in a folded manner, and then DNA, it should fold on protein for becoming more compact and systematic. Second event is formation of large intracellular membrane bound organelles. And third event is formation of the autotrophic cells or photosynthetic cells and primitive eukaryotic cells. So this is how the first cell it appeared, and like this we have completed this topic, that is the origin of cell and unicellular organism. So if you like my video, please give it a thumbs up. Please subscribe our channel, that is Let's Crack CSIR-UGC NET. And for getting all the notification, please hit the bell icon. And for getting a 10% discount in the subscription, in the plus subscription, which can be very much helpful for you to qualify the examination, please use my referral code, that is FP01. So by using my referral code, you will be eligible to get a 10% discount. So do not lose this opportunity of getting a 10% discount. So this is it for today's class. We'll be meeting again tomorrow with some interesting topic. So this is it for now. Have a very good day. Bye from my side.